Thank you very much, Lani, for the invitation to come and speak, and apologies for the flight delay. So what I would like to do today is provide some brief background for a few minutes about where the field has come over the last few years identifying the genetic basis of ALL, and present a couple of stories, one published and one not yet published, showing you specifically some of the insights in high-risk and relapsed ALL. So as you know very well, ALL remains a leading cause of cancer-related death in young people, particularly in adolescents and young adults, where the genetic basis is much less well understood than in young children. Much of our knowledge about the genetic basis of ALL has been from the study of young children and children prior to adolescence. And these studies have really completely reclassified ALL. It's identified new subtypes of leukemia. These studies have identified the constellations of structural and sequence changes that act together to induce leukemia. And importantly, they've begun to identify new diagnostic and therapeutic targets, either by specific mutations or by pathway-directed approaches, as you've just heard. So until about a decade ago, this pie chart really illustrates how we approach subclassification at a genetic level of childhood ALL, where, as you know, the majority of cases are of B lineage and a smaller proportion are of T-cell origin, shown in blue and red respectively, and the majority, but not all, have a sentinel chromosomal change, such as a aneuploidy or a chromosomal rearrangement. And these rearrangements are very important in deregulating hematopoietic transcription factors, tyrosine kinases, and epigenetic regulators, but alone are usually not enough to cause leukemia. And we now know that multiple pathways are mutated in the majority of patients. Importantly, and again this is from childhood leukemia, approximately a quarter of cases didn't have one of these genetic changes, and the frequency of these genetic unknowns rises with increasing age. So arguably, the majority of what we now know about the genome of ALL has actually been derived from pre-sequencing studies, and what I've called here first-generation genomics, which have used microarray approaches to identify DNA copy number alterations, as well as profiling gene expression patterns, coupled with fairly limited but important results from candidate gene sequencing. So what have these studies shown us? Well, one of the first hits that you've heard introduced by Rick is that the majority of B lineage ALL cases have one or more lesions that perturb normal B cell maturation. And in fact, the most common lesion is PAX5, not EPOR, but EPOR and other genes are also involved. As you've already heard, these hits are very important because they occur at logical stages that correlate with the arrest in B cell maturation that's so often a hallmark of B cell ALL. And in contrast, you rarely, if ever, see the transcription factors that control the later stages of lymphoid maturation mutated in B-ALL. But in addition, there are a number of other genes and pathways that are mutated, and the repertoire of these individual gene changes is expanding almost week by week. But many of them now fall into logical groups, such as tumour suppressor genes, such as the INC4ARF complex, P53, particularly in relapse leukemia, many proliferative genetic changes in RAS, cytokine receptor and kinase signaling pathways that I'll come to in a moment, as well as some other changes that we don't understand mechanistically well yet at all, particularly involving transcriptional regulators and transcriptional coactivators. And an emerging theme is that genetic changes perturbing epigenetic regulation and histone modification are also important, including at relapse, and I'll come back to that later in the talk. Several of these have already transitioned to the clinic. Several groups use EPOR profiling to risk stratify patients at diagnosis, and some are logical candidates for therapeutic intervention, such as JAK inhibitors and other kinase inhibitors. And we've now been able to reclassify ALL by identifying new subtypes of leukemia. For B-ALL here shown in yellow, where there are several important subgroups that we and others have identified, such as ERG-deregulated leukemia, PH-like or BCR-ABL-like leukemia, and others as well. So moving on to sequencing study, and this does pour justice to a number of studies from multiple groups over the last 3 or 4 years. These studies are really continuing to lift the veil on the leukemia genome. Our initial studies at St Jude in a genome sequencing project of childhood cancer, as well as other studies, have initially focused on high-risk subtypes of childhood blood and solid tumours. And so the first that we studied was this interesting subtype of aggressive T-cell leukemia, early T-cell precursor ALL, where based on a fairly homogeneous gene expression profile, we expected to find one or more undiscovered genetic changes. But in fact, we found multiple pathways harbouring multiple mutations, including transcriptional regulation, signalling and epigenetic regulation. We focused on an interesting subtype that's grossly aneuploid with multiple whole chromosomal losses, and identified again the same themes or pathways harbouring mutations, but here the genetic changes were very different. And so for the first time we were finding alterations in other members of the EPOR gene family, and we're now starting to understand how they cooperate in leukemogenesis. This was also clinically important because we found many of these cases are actually manifestations of Li-Fraumeni syndrome by having inherited P53 mutations. And as I'll come to, PH-like ALL is perhaps one of the most clinically important and exciting lesions. It's also very common, and I'll come back to the sequencing basis of the sequence studies defining the basis of this in a moment. And one other point that I won't go into further is that studies are identifying a range of different common and also uncommon genetic changes, either subtle inherited variations or highly deleterious mutations that to various levels influence the risk of developing ALL. And in fact, studies from St Jude have suggested that over 10% of children have some sort of deleterious mutation in their genome. So for the rest of the talk, as mentioned, I wanted to focus on two areas. The first is how we led to the identification and subsequent characterisation of PH-like ALL, and how we're attempting to transition those findings into the clinic. And then, if time permits, the way that we're using sequencing to understand how the leukemia genome evolves during the course of therapy, and how that also might be clinically important for diagnosis, monitoring, and also new therapeutic approaches. So I won't belabour this, but as you've heard, the pH positive or the pH chromosome encoding BCR-ABL is a hallmark of both ALL, which is very aggressive, and CML, which is more indolent and responds better to TKI monotherapy, but which can also progress over time to acute leukemia, either AML or ALL. And when we started doing microarray-based profiling, we obtained as many pH-positive leukemias of both flavours as we could to look for the genetic changes that distinguished them. And as you've heard, the commonest one that was found was alterations of ICROS, either sequence mutations, but more commonly deletions, either loss of function or dominant negative deletions. At the time, and this is going back now to about 2007, 2008, at St Jude we began collaborating with the Children's Oncology Group, who had a childhood equivalent of the TCGA called the Target Project, which was to focus on pediatric cancers by sequencing profiling, and one of the first entities to be studied was high-risk B progenitor ALL, and specifically studying cohorts of cases that were high risk and lacked many of the sentinel chromosomal rearrangements that are otherwise common in ALL. And one of the first findings from this study was the identification of a subtype of pH-negative high-risk leukemia cases that often had an alteration of ICROS and also experienced very poor outcome. These cases have a transcriptional signature like pH-positive leukemia. We identified this using gene set enrichment analysis, and the Dutch identified this using hierarchical clustering, but the finding was essentially the same. There's this group of cases that has a similar gene expression profile, but it's pH-negative. And so clearly the two implications of this would be that it's ICROS itself that's contributing to an immature gene expression signature or other gene expression signatures, and we now know that's the case, but also that there are other underlying lesions driving kinase signalling, and we now know that that is almost always the case as well. If we move forward now, we've just published a large study looking across the age range from children just beyond infancy up to the age of 39, using initially microarray gene expression profiling and subsequent genomic characterisation and sequencing to define the prevalence across that age spectrum of PH-like ALL and to look at outcome and the underlying genetic lesions. So this study included over 1,700 ALL cases, B progenitor ALL cases from St Jude, the COG and the adult cooperative groups. And the key finding in terms of frequency was that in childhood standard risk leukemia, PH-like ALL is much more common than BCR-ABL positive leukemia, and the frequency rises with increasing risk features and also with age. Data is starting to emerge about the prevalence of PH-like ALL beyond young adulthood, but those studies at the moment are ongoing. But you'll note that at least half of patients are potentially amenable to intervention with a tyrosine kinase inhibitor, at least in the older age groups. So the genetics are somewhat complex, but just to spend a couple of slides going through them, they can be grouped into several logical pathways or groups. Quite consistently across the age range, approximately half of PH-like ALL cases have alterations of one signalling pathway, and that is signalling through cytokine receptor-like factor 2 or CRLF2. So this is a cytokine receptor that forms a heterodimer with interleukin-7 receptor alpha, and often these cases also have a mutation of the Janus kinase genes, JAK1, or more commonly JAK2. The rearrangement is either a mixed-style rearrangement of CRLF2 to the IGH locus or a focal deletion immediately upstream of CRLF2 that results in a novel fusion transcript. And both of these events deregulate expression of the intact CRLF2 protein. This can be detected by molecular approaches very conveniently, by FISH as shown here, and ultimately by flow cytometry, as these rearranged cases have increased cell surface expression of CRLF2, and so this has already been implemented as a diagnostic test. The remaining cases are more diverse and somewhat more complex. Of the remainder, about 80% have one of a range of different rearrangements that target kinases or cytokine receptors, and so far over 13 have been identified and over 30 different individual rearrangements. Approximately 10% have non-rearrangement alterations, some of which I'll discuss in a moment, either sequence mutations or structural alterations that also deregulate kinase signaling, most commonly JAK-STAT signaling, but some other pathways are involved as well. And again, this recurring theme that the majority of cases have alterations of lymphoid transcription factors, most commonly Ikaros, but sometimes others instead or indeed in addition to alteration of Ikaros. Our initial studies, again as part of the Children's Oncology Group target initiative, was RNA sequencing of 15 PH-like ALL cases. This doesn't sound like very much, but this was a few years ago when this was very new in terms of investigating the genetic basis of cancer, and we were just dipping our toe into the water of doing RNA sequencing. And we started with this because it was shown to be very fruitful in identifying chimeric fusions, and we hypothesised there might be such fusions that are substituting or phenocopying for BCR-ABL. So the 15 cases chosen, 3 of which had a CRLF2 rearrangement. One of the reasons we selected those was to see if we could find a lesion that was substituting for JAK mutations, which are not present in all of those cases. But the remainder did not have a JAK2 rearrangement. The remainder did not have a CRLF2 rearrangement, but they did have a range of different fusions targeting different tyrosine kinases or cytokine receptors. PDGF receptor beta, JAK2, ABL itself, a case with an erythropoietin receptor rearrangement, and you'll note a diverse range of fusion partners. You can see here that there were a couple of cases that did not have a fusion, and in those whole genome sequencing was used and identified some of these initial sequence and structural variants that also trigger kinase signaling, and that's an important theme, particularly as we consider how we implement this diagnostically. So if we come back to the present and our completed study, so after identifying these PH-like cases across this cohort and then taking out the CRLF2 rearranged cases which were identified by candidate assays or FISH, we then subjected the remaining cases, over 150, to some level of genomic profiling, most commonly RNA sequencing, but also in a small number of cases, a smaller number of cases, exome and or whole genome sequencing to try and comprehensively define the genomic landscape of these cases. After identifying new lesions such as fusions, we then returned to larger recurrence cohorts to ask the question, were these rearrangements selectively present in PH-like ALL, which almost always they are. So this is a rather detailed heat map attempting to show the kinase activation heat map landscape of these PH-like ALL cases. And we've attempted here to group them into both the class of kinase signalling and also the nature of alteration. And I'll go through these example by example, but one could see once we'd now obtained data from many patients that different themes were emerging in terms of the type of kinase and the type of alteration. So the first and arguably the most immediately important group is what we've called ABLE class rearrangements, because they involve kinases that we now know are quite sensitive to classic ABLE inhibitors such as dasatinib and imatinib. So multiple rearrangements involving ABLE itself, but rearrangements of ABLE2, the homologue, also known as ARG or ABLE-related gene, rearrangements, surprisingly, of CSF1R or the macrophage colony stimulating factor receptor, and also a number of rearrangements involving PDGF receptor beta, which are also interesting because in some studies they appear to be quite strongly associated with induction failure and very aggressive disease. And below you can see in blue the multiple fusion partners that are involved. I've just shown a couple of examples of the protein domain structures to make a couple of important points. That is, the kinase is always at the 3' end of the fusion protein. The catalytic kinase domain is always preserved intact. And also of biological interest to us is that the fusion partner likely has many important roles, including deregulation of the transcription factor that may be fused, such as EBF1, and other ways of causing mischief, such as resulting in constitutive dimerization or subcellular mislocalization. We see similar themes for a second class of rearrangements, and that is the JAK2 and EPO rearrangements. JAK2 rearrangements are interesting because they're rather uncommon in young children, but they become progressively more frequent with increasing age. They were particularly common in the AYA cases obtained from the ECOG cohort that you've heard referred to already. And also some rearrangements were identified of the erythropoietin receptor locus that I'll come back to in a moment. Then we turn back to CRLF2 rearrangements, and just to make one point about this, we didn't sequence all of these cases, but we did select some, again to try and get to the bottom of this question as to what were the other lesions that might be substituting for JAK2 point mutations in these cases. And so you can see here there are the cases here with JAK2 sequence mutations, but some don't. But we were now starting to see additional changes that also appear to act in concert with CRLF2 rearrangements, such as point mutations in the IL-7 receptor, FLT3, and a negative regulatory gene called SH2B3 or LINK. It's important to note that not all cases do have rearrangements. So there is an important group of cases that have no rearrangement, but they have, they're characterised by sequence mutations or less commonly deletions targeting JAK-STAT signalling and also FLT3 signalling, or less commonly the other members of the Janus kinase family such as JAK3 and TYK2. These cases may not have rearrangements targeting kinase signalling, but they certainly do have rearrangements, although here the rearrangements are now targeting transcriptional regulation, EBF, ERG, ETV6 or TEL, but a number of new fusions, and also a number of rearrangements targeting epigenetic modifiers such as ASXL1, CBP, and others as well. So again, it just speaks to this theme that the pathways are the same across patients, but the mechanism of alteration differs between different cases. So I just wanted to show one example of how informative and elegant the genomic data can be, and that is to spend a couple of slides on the rearrangements of the EPO locus in ALL. So we identified one case with what appeared to be an EPO rearrangement in our first study from RNA sequencing, with the top panel showing the marked overexpression from digital gene expression across the EPO locus. At the time, we didn't have very robust algorithms for identifying these often complex rearrangements into repetitive areas, and this required some manual labour. But eventually, Jingwei Zhang, a key collaborator in this group in computational biology now at St Jude, was able to manually map these reads and show that there was a rearrangement just of the 7 kilobase region of the EPO locus into the immunoglobulin enhancer region. So not a reciprocal rearrangement, but a cryptic insertion that would otherwise be missed by FISH. And at the moment we set it aside and paid no further attention, But we now know that these rearrangements are recurrent. They're restricted to pH-like ALL. They involve either the IGH region or immunoglobulin kappa, and they always truncate the last exon, the cytoplasmic tail of the erythropoietin receptor locus. So if you see here from this read coverage heat map, this is the 5' end, this is the 3', this abrupt truncation in coverage halfway through the last exon. So what might this do? Well, the cytoplasmic tail of the EPO receptor, normally a homodimer that binds EPO, has a number of different tyrosine residues, the most proximal of which is indispensable for normal signalling, but the remainder of which either help with signalling or actually help with negative regulation and internalization of the receptor. And each of these rearrangements and truncations spare this first tyrosine, but most of them then remove most or all of the other distal tyrosines. This became even more interesting because as we started looking at the literature, we observed that these truncations in their location are essentially identical to the sequence mutations that are inherited in primary familial and congenital polycythemia, which is an inherited condition resulting in erythrocytosis and EPO hypersensitivity. And that's shown here where you can see now in green the position of our rearrangement truncations and in blue the point mutations that you see in PFCP. And so we would predict that these truncations would also truncate the receptor and lead to augmented JAK-STAT signalling. We now know that's the case. To summarise a lot of work on one slide here, we've shown that these truncated EPO isoforms result in retention of the receptor on the cell surface and delayed degradation. They result in markedly augmented signalling through JAK-STAT, here shown as phosphosignalling for STAT5. And primary leukemic cells are quite sensitive to a JAK2 inhibitor, ruxolitinib, ex vivo. These are cells harvested from a xenograft. The same tumour cells are quite sensitive in vivo in a xenograft model of EPO-rearranged leukemia. You can see this by the imaging on the left or by looking at the spleen weights after harvesting. So this appears to be a novel therapeutic approach to this specific class of fusions. As time is short, I won't go into detail about the functional analyses that we've done, but the goals here were to show in a range of different fusions that we'd identified, particularly for the new kinase partners, that they did activate the signalling pathway that was predicted and that was sensitive to logical TKI therapy. These are very standard cellular proliferation assays using a growth factor-dependent cell line BAF3, showing conferral of factor-independent cell proliferation that's quite sensitive here for the ABL class fusions to dasatinib. And we can see here shutdown of the signalling pathways of these cells, here now looking at ARF-null pre-B cells in ABL class fusions, one example here shown with shutdown of signalling to dasatinib, but not to the JAK2 inhibitor ruxolitinib. And we've now established a large number of xenografts here. We've engrafted over 50 tumours from over 70 that we've attempted, and we're pushing ahead to try and do in vivo preclinical studies to try and take sentinel tumours, again to demonstrate that primary human leukemic cells are also sensitive. And I think remarkably for such an aggressive disease that we would never anticipate treating with tyrosine kinase inhibitor monotherapy, these tumours are often quite exquisitely sensitive to TKI therapy in vivo in a mouse. They're not eradicated. In fact, some do eventually break through after prolonged treatment, but they're often potently suppressed for a long period of time with the TKI alone. So these findings really provide the rationale for testing efficacy in patients, but how might we group them together into different therapeutic approaches? So I mentioned there are 4 groups of kinases that are now known to be logical targets for dasatinib or similar agents. There are several targets potentially for JAK2 inhibition that may be variably sensitive to monotherapy with this inhibitor. And then there are a smaller number of less common or even singleton fusions, but that also have available approved agents that could potentially target these tumours. An interesting one we've spent a bit of time looking at, only a single case so far, that we've shown a rearrangement of NTRK3 that's seen in other tumours that's highly sensitive to crizotinib. We now know that there are multiple patients that have responded, that have had aggressive or indeed refractory disease. And I won't go into the details, but in the paper we published recently and prior reports, several groups now have reported multiple cases, particularly cases with ABL class fusions with refractory disease that have had remarkable rapid and sustained responses. So how might one implement this into a clinical approach? Well, one can envisage several levels of complexity in sequencing. So perhaps the most luxurious one would be what St Jude has instituted, which is by early next year, all children with all tumours that are newly diagnosed will have comprehensive whole genome, exome and transcriptome sequencing. That's clearly not feasible, at least not at the moment, for very large studies. So there are other approaches that are possible, such as targeted sequencing, which is becoming increasingly widely used, although one can perhaps anticipate this will be somewhat complex because of the genomic heterogeneity. And the other end of the spectrum, the Children's Oncology Group is planning a study that will also open in the near future of a national study of PH-like ALL that will use a tiered approach. And just showing the basics of this, the goal here is to exclude other subtypes initially that we know are never PH-like, and of the remaining cases to screen them with a targeted gene expression array that was developed by Cheryl Willman's group at the University of New Mexico, just a small number of genes to identify the PH-like phenotype. We then use candidate assays to screen for the lesions that we know of, and then the remainder undergo sequencing where no lesion is found. And so, as mentioned initially, the goal is to commence dasatinib in combination with chemotherapy for the ABL class fusions, and then eventually our hope is that additional targeted agents will be used in the remaining cases. So to conclude this part of the talk, PH-like ALL is important, it's common, its frequency increases with increasing age. The genetics are complex and they continue to become more complex as we study more cases, but there are a relatively small number of signalling pathways that are logical candidates for targeted intervention. And although the genetics are complex, there now are good tests, some of which are already clinically available, to both identify and test for the underlying genetic lesions in PH-like ALL. So I think I have a few minutes remaining. Just in the last few minutes, I wanted to show some unpublished data, again from a collaborative project with the Children's Oncology Group, showing how there's much more to the leukemia genome than just the nature of the studies I've shown you already, which is really profiling the bulk leukemic cell population. So we've known that the ALL genome is not static, it evolves from diagnosis to relapse. That's been known from cytogenetics for many years. And we and others showed some years ago using SNP arrays that the majority of diagnosis and relapse clones from a given patient share a common clonal origin, and that was shown by various levels of alteration. We showed that DNA copy number alterations, that's deletions and gains, show marked evolution from diagnosis to relapse. And we could also use backtracking in many cases, so molecular backtracking, to show low levels of the relapse clone being present at diagnosis. So that allowed us to put together this schema in which, just using very limited sequencing in conjunction with copy number analysis, only a minority of cases showed a completely unrelated second leukemia at relapse. Similarly, there were very few that showed no evidence of change from diagnosis. But the remainder either have clonal evolution or they have a more complex pattern, and this is the most common pattern, where we have a pre-leukemic clone that then undergoes divergent development or ancestry or evolution to propagate different clones that are present at diagnosis and relapse. Now, in recent years, we have begun to identify the specific genetic changes that facilitate emergence of this relapse clone and that have direct implications for the mechanism of resistance and potential for targeting. So one that you've heard a lot about is EPOR, which may emerge as a new lesion at relapse. Another is a lesion we identified called mutations in CREBVP or CREB binding protein that we now know directly facilitates resistance to steroids, obviously a mainstay of ALL therapy. And two groups identified mutations in a nucleoside metabolizing gene called NT5C2, again providing another mechanism for resistance. But questions remain unchanged, so how can we comprehensively examine how clones rise and fall during disease progression and the relationship of mutations in that process? And also, we've noted, and others have noted, that some patients actually have multiple clones with subclonal mutations in the same pathway. How does that work, and how does that influence the progression from diagnosis to relapse? So in a pilot study with the COG, 20 cases were subjected to relatively deep sequencing, not ultra-deep sequencing, but relatively deep exome sequencing. The majority of cases also had whole genome sequencing, as well as SNP array analysis for copy number alterations. And importantly, all of the mutations were then revalidated by taking all putative sites of mutation and then sequencing quite deeply with an orthogonal approach at each time point. So in these couple of heat maps, this just shows, admittedly in a bit of a busy slide, again a way of trying to group together the relapsed enriched lesions. And again, they fall into the same pathways as we've seen before: RAS signaling, JAK signaling, lymphoid maturation and cell cycle arrest, as well as purine metabolism as mentioned before. A new theme that's emerged from this study and from other groups such as Scott Armstrong's group is that an increasing number of chromatin-modifying genes are also emerging as new hits at the time of relapse, so important both for mechanism and also potential therapeutic targeting. And many of these are shown here. I mentioned CBP, but a number of other important genes have also been identified as recurring targets of mutation. And so then another question was addressing this question of multiple subclonal mutations. So an orthodox view is that if you have a subclonal mutation, it's probably a passenger and it's probably not important as a driver in leukemogenesis. Well, this study identified that at least half the cases had at least two, if not more, mutations in one or more key pathways. The same key pathways that we'd studied before, often sequence and structural mutations in the same gene, and remarkably, often mutations targeting the very same gene or even residue in some cases. One of which is shown here. So this is JAK2, as I'd mentioned before, with this hotspot of mutations at a residue in the pseudokinase domain R683. So this case had three mutations. This is the first one. This is the reads for the wild type allele, and the mutant allele shown at diagnosis, at remission, and then relapse. So just a couple of reads at diagnosis, then completely gone. A second mutation at low level at diagnosis that then became predominant at relapse, and a third mutation that was predominant at diagnosis, but then completely lost at the time of relapse. And so if one then pulls together all of these data for every mutation, one can refine this sort of schema which has been widely used, where people attempt to tease out different subclones at diagnosis and relapse with much more sensitive mutational data to get a more detailed picture of both the large clones and also the small clones that are present at each time point, at diagnosis, at relapse, or at both time points as well. So using this information, we can actually count the number of clones and then chart their rise and fall. And so this is the same example that had these three different JAK mutations at diagnosis, two of which are lost, but then one of which wins out and survives through to propagate relapse. And then this clone acquires additional mutations, this one here, an NT5C2 mutation, very important in resistance, as well as others to propagate the predominant clone. One can then take all of this information, the number of clones, their size, and the number of mutations per clone to count the number of clones and characterise them both at diagnosis and relapse. And the final piece of data I'll show, and the final point, is that in virtually every case, relapse is arising from a minor clone at the time of diagnosis. And these cells acquire one or more of these mutations that facilitate resistance to therapy and then emerge at relapse. Occasionally, we do see relapse arising from a major clone at diagnosis, but then this has an explanation as well. These cells often have the mutations that we know are enriched in other relapse cases and facilitate resistance, such as P53 or an epigenetic regulator. And so there I'll stop. So just to conclude this part, yes, we've confirmed that multiclonality is a very important and common event present in most cases at the time of diagnosis. Multiple genetic changes involving the same gene or pathway are more common than we previously identified, and we've now expanded the number of relapsed, or a number of pathways and genes that appear to facilitate relapse. What's the clinical relevance? Well, there are at least 2 points that I just finish on. One is that, yes, clearly some of these genes have important implications as new targets for therapy, but also we know that many of these mutations that end up being present in the relapse predominant clone are actually present relatively early during disease evolution. And so as we start to think about how we implement clinical genome sequencing and monitoring of minimal residual disease, strategies to identify these low-level mutations will clearly be important. And my final point is that I've spent a lot of time talking about PH-like ALL, and it's a popular area of investigation at the moment, but there's still a lot we don't know. If we look at these pie charts now of children, adolescents and young adults, we still have a lot of work to do, with up to 30% of cases, depending on age, still lacking a known genetic lesion, and many of our relapses still occur in these populations. So clearly a fertile ground for ongoing work. So I'd thank several people from my group in St Jude, particularly Catherine Roberts, who did much of the PH-like work, and Ilaria Yakabuchi, that did the EPOR receptor work, Jingwei Zhang, who led the bioinformatics, and Yongjin Li, who did the analysis of the PH-like work. And the many collaborators that have contributed to this work, most importantly, colleagues from the Children's Oncology Group and that consortium, the Target Group, as well as the NCI for funding the Target project. And there I'll stop. And if there is any time, I'd be happy to take any questions.